Greetings students, and welcome to the lecture on synthetic biology. Synthetic biology is a discipline which focuses on the development of synthetic genes. These synthetic genes can be applied for multiple experiments and can also be utilized for downstream applications such as the production of vaccines and recombinant proteins. In this lecture, we will be studying different elements of synthetic biology, and I will introduce you to what is termed as the parts registry. The summary of this lecture is as follows. I will introduce you to synthetic biology, the parts registry, the components of the system, devices, applications of the polymerase chain reaction in synthetic biology, Gibson assembly and assembly PCR. We will commence with the key aspects of synthetic biology, and we will then delve into the different terminologies associated with synthetic biology. The basic aspect of synthetic biology is the parts registry. So the parts registry comprises different parts which can be assembled in order to form functional genomic assemblies. The different parts are termed as components, and a device is a part which is developed by combining one or more components. When we do experiments involving synthetic biology, we have to be aware of the PCR, which involves Gibson assembly as well as assembly PCR. These are the two tools which are used to engineer DNA molecules. What exactly is synthetic biology? Synthetic biology combines biology and engineering to create new biological systems which currently do not exist in nature. So what essentially synthetic biology refers to is the engineering of genes to create systems. And in this case, we look at the genes in terms of their functionality. In order to assemble genes, the polymerase chain reaction is employed, and we use a specific method known as Gibson assembly to assemble genes. Synthetic genes can also be created using gene synthesis approaches, in which case you can combine different oligonucleotides to create large genes. Some of the examples of synthetic biology are bacteria that generate fluorescence in response to a biochemical signal, and this is essentially what is termed as a biosensor. Bacteria can be engineered to produce specific visual or optical cues in response to chemical receptors or chemical signals in the environment, and this forms the components of biosensors. Viruses can be recombined to study viral replication and infection. So when we utilize synthetic biology, we can combine different viral elements to create novel viruses, which can also be used for engineering applications. Bacterial cells can be engineered to secrete proteins in response to environmental cues. Now, this is a very useful tool when you do recombinant protein expression, as the bacterial cells can be engineered to secrete proteins in response to a specific cue, such as the addition of a biomolecule, and this is utilized in the production of recombinant proteins. And bacteria can also be engineered to switch on or regulate specific biosynthetic pathways in response to stress. All of these represent elements which incorporate synthetic biology into their basic construction and deployment. Now, in synthetic biology, we use certain terminologies. Just as you have physics and electronics, and in the electronic circuitry you have components such as resistors, transistors, capacitors, and so on. In synthetic biology, we use specific terminologies to address molecular biology components. And in this case, we have the parts and devices. So the parts are basically promoters and ribosome binding sites. Devices can be in the form of reporters, inverters, and sensors, and functions involve biosynthesis, conjugation, and cell-to-cell signaling. So we will look at this in detail. To begin with, you have to understand the structure of a gene. So this is a schematic representation of a gene. Now in this case, you have the 5' and the 3' end, and then you have specific parts. The gene comprises a promoter binding site, a ribosome binding site, and you have the protein coding region. So if you look at this, you have a ribosome binding site here. You have the start and stop codons, and then you have the gene of interest itself, which is the open reading frame. Now, if you want to control this gene, you can introduce additional regulatory elements such as promoters at this end, and this will in turn regulate this gene. For instance, if the gene was encoding a fluorescent protein and you wanted to ensure that this gene was induced in the presence of sugar, you can introduce a promoter binding site here. And this promoter binding site is specifically regulated by the presence or absence of sugar molecules in the bacterial culture medium. So when the sugar molecule is introduced into the bacterial culture medium, this gene will be turned on. In the absence of the sugar molecule, the gene will be in the off state. So this is just one example whereby you can utilize synthetic biology in terms of the sensing abilities. Now, genes can be assembled using something known as Gibson assembly. So in Gibson assembly, we basically combine genes using a polymerase chain reaction. Now, I have depicted these genes on plasmids. So the genes are generally stored on plasmids as PCR products tend to degrade in cultures. So we have our genes. So we have these 4 genes, and you can store these genes in glycerol stock in bacterial colonies, and you can then assemble them when you need them. So this diagram depicts the different combinations of genes which can be assembled using Gibson assembly or an assembly PCR. Now, the parts registry is basically an assembly standard. So when you have an assembly standard, this generally refers to a method or a standard which can be replicated across labs. For instance, if your lab is located in Malaysia and you're sharing a gene or a component with a lab which is located in Germany, the assembly standard will remain the same if you adopt the Biobrick assembly standard. And what this Biobrick assembly standard refers to is the restriction sites on either side of the gene of interest. So this is your gene of interest, and this is the restriction sites, or these are the restriction sites on either side of this gene. Now, what happens is that when you transfer this gene from one country to another or one laboratory to another, you always know which is the restriction site. So for instance, if I wanted to excise this gene, I can use any one of these combinations of restriction enzymes. For example, use XBA1 in combination with NOT1, and then you have a double digest, and you can excise this gene and introduce it into another vector. So this Biobrick assembly standard facilitates the standardization of components across laboratories. We will now delve into the different components. So as you know, a promoter is a DNA sequence that can recruit transcriptional machinery and lead to transcription of the downstream DNA sequence. Now there are a variety of promoters, and they are induced by different kinds of cues, environmental cues, and we have strong and weak promoters. We have constitutive and inducible promoters. Now, inducible promoters are generally set into play or set into motion by the introduction of an inducing agent, whereas the constitutive promoters are responsible for the expression of the gene at a baseline level. We also have strong and weak promoters. Strong promoters lead to a high copy number of the transcript, whereas a weak promoter will lead to a low copy number of that particular transcript. Now, when we look at the synthetic biology terminologies, the symbol for the promoter is this particular symbol. This is an arrow and it's indicated as such. So we have different kinds of promoters. So as I mentioned earlier, we have the constitutive promoter, which is responsible for the steady expression of the gene with which it is associated. We have the positively regulated promoters, which are regulated by the introduction of a specific regulator, such as a sugar molecule or maybe a metal ion. We have the negatively regulated promoters, which are represented by this symbol. In this case, the promoter is responsible for turning off the gene in the presence of the specific environmental cue or the specific inducer. And we also have multi-regulated promoters, which may be turned on by one cue and then turned off by another cue. So this is a representation of the parts and the components associated with synthetic biology. Regulated promoters are specific sequences which can be utilized to turn on or turn off specific genes. In addition to regulating single genes, they can also operate as promoters for multiple genes, and they can also serve as feedback regulators. Now, these kinds of promoters are complex and sometimes they are associated with multiple proteins, and their specific regulation is termed as regulated promoters. Now, promoters can be validated. The promoter can be validated using a green fluorescent protein molecule. So you have a promoter at the 5' end of the gene, and then you fuse it to a green fluorescent protein. And you can regulate this promoter by measuring the intensity of these particular fluorescence in a bacterial culture system. So if you have an intense expression of this gene as indicated by the optical signal, it indicates that the promoter is strong. However, if you have a weak expression of this gene, there is less fluorescence, and this is indicative of a weak promoter. Now, validation of promoters is essential because when you utilize a promoter for industrial expression of certain recombinant genes, you must assess the promoter strength. Weak promoters are not good for production systems, as you will be spending a large amount of energy to cultivate the bacterium and obtaining a very low product yield. So promoters are generally regulated by using IPTG, and IPTG regulated promoters can also be evaluated based on their fluorescence signal. So this is an example of a promoter sequence. So in this state, the bacteria E. coli carrying the promoter do not express the gene. However, after induction with IPTG, you have an induction of the gene. Now you will note that on this plate you will see some few dots of color. So this is indicating expression, and this may be a leaky promoter, which means that the promoter is not very tightly regulated, and in some cases it has expressed the green fluorescent protein. This is an example of a single colony carrying a gene which has been induced using IPTG, and you can clearly see the fluorescent signal associated with this colony. Now, promoter binding sites are designed by annealing two oligonucleotides. If you have done PCR and carried out PCR reactions in the lab, you must be familiar with the concept of the primer, which is essentially an oligonucleotide. Now, when you want to construct a promoter binding site, you can combine one or more primers, and this represents the complementary region. So you can design these primers so as to have an overlap. Subsequent to that, in a polymerase chain reaction, they will produce a PCR product which is representative of a promoter. Now these promoter sequences can be obtained from the NCBI gene bank, as most of the promoters have been well documented. So this is basically how you design an oligo. So this is a forward oligo and a reverse oligo, and they will have an overlap, and you will get a resultant promoter sequence as in this sequence over here. In addition to the promoter binding site, you can design the ribosome binding site in a similar manner, and you require ribosome binding site in between the promoter binding site and the gene of interest in order to ensure that it is transcribed and subsequently translated. This is a typical ribosome binding site sequence, which is about 6 nucleotides upstream of the start codon. So this is your start codon, and this is your ribosome binding site. So the ribosome will essentially bind here, and this will lead to the translation of the protein. So these are the ribosome binding sites, and you can see that they are weak and there are also very strong sites. And we have what is known as the Sheen-Dalgarno sequence, in which case you have this particular sequence GGAG, and this is basically associated with the start codon. So when you view this Sheen-Dalgarno sequence, which is upstream of the gene of interest, you can basically understand that there is a ribosome binding site associated with that particular gene. So all you need to do is look at the AUG, the start codon, and then look upstream and look for this particular sequence which represents a ribosome binding site, or an RBS. So in the E. coli itself, you have the ribosome binding sites, and this is a schematic representation of the gene in which you have ATG as the dominant start codon. You can also have GTG in some cases. So in this case, the height of this particular alphabet indicates the probability of that site being present in the start codon. So in this case, it's generally ATG, and in very rare cases, it is GTG. In addition to ribosome binding sites, there are also elements known as riboswitches. These riboswitches perform regulatory functions by folding into specific configurations. In this case, this hairpin configuration can turn on or turn off expression of the downstream gene. Now this switch can be regulated by both biomolecules as well as temperature. Let us look at the riboswitch in terms of its configuration. So now you have a configuration which is off. So what has essentially happened is your RNA molecule has formed a secondary structure, and this secondary structure prevents the binding of this particular ribosome. Now, because the ribosome cannot bind, there is no translation of this protein. However, when this configuration is resolved, so you have no more secondary structure, the protein is translated from this particular ribosome binding point. So you'll have the ribosome binding to the mRNA, and you'll have the generation or the translation of this protein. Now this particular configuration can be regulated by both temperature or by other biomolecules. And riboswitches are essentially genetic elements which can be used to turn on and turn off gene expression by the application of certain cues to regulate secondary structure. So we look back at the domain itself. So when we look at a protein domain, we note that we have a ribosome binding site, a start codon, and then you have functional domains within the protein itself. So we have the head domain, the tail domain, and the internal domain. Now you have to understand this from the perspective of a gene which encodes an enzyme. in the enzyme coding genes, the internal domain represents the catalytic domain, and this catalytic domain is enclosed between a head and a tail domain, and the enzyme activity is regulated by the internal domain itself. So this is just one example of a protein domain as indicated in a synthetic biology representation. Protein coding sequences essentially are open reading frames, so you can obtain a protein coding region from a NCBI gene bank, and you can construct a fusion with protein itself, the protein coding region, the promoter, and other regulatory elements. And in this case, we use the word chassis, which is generally associated with engineering parts. So the chassis is basically a host which is utilized to express that particular protein. So as you can note, there are many terms which are utilized in synthetic biology, and it basically looks more like engineering in terms of its utilization of terminologies, as it uses terms such as chassis, the functional parts, functional domains, as well as elements such as the regulatory elements. Finally, you have the part which is known as a terminator. So terminators are basically representations or parts which turn off the gene expression at the end of our RNA molecule. So when a ribosome travels along the gene, it will reach a point at which it encounters this stem loop structure, and at this point the ribosome will basically disassociate itself from the mRNA molecule, which indicates the termination of translation. So there are row-dependent palindromic sequences and row-independent sequences. So these are both representations of the terminator. So terminators are generally located at the 3' end of genes. In the case of row-dependent terminators, we have palindromic sequences. So this will form a stem loop structure as indicated over here. In the case of row-independent, we do not have palindromic sequences. There may be variations in the sequences. Moving on to synthetic biology, we use the term composite parts. Now, composite parts are basically implying that more than one element has been joined together. This may be in the form of a gene which has been fused to another gene, a multiple set of genes which is regulated by a single promoter such as an operon, or other components. So we utilize these genes to carry out specific functionalities as defined by the engineering constraints. We move on now to specific techniques associated with synthetic biology. And there are two basic techniques. The first one is Gibson assembly, and the second one is assembly PCR. Among this, Gibson assembly is the more advanced technique which is routinely used in synthetic biology laboratories. So Gibson assembly is basically a DNA ligation technique which was developed by Dan Gibson at the J. Craig Venter Institute in 2009. So it utilizes 3 enzymes to ligate 1, 2, or 3 sequences with an overlapping end. Now it's very important to note that when you want to join or ligate 2 sequences, there should be overlapping ends. This is because of the enzyme chemistry which is utilized in a Gibson assembly reaction. Let us look at Gibson assembly in detail. So these are the enzymes which are utilized in Gibson assembly. You have a T5 exonuclease, and as the name suggests, it is an enzyme which degrades DNA at the terminal 5' and 3' ends. We have Taq ligase, which will not ligate any sequence which has a single mismatch. So what this basically means is that if you have an assembly PCR And if you have taq ligase, there will be no ligation in case of an error. And this is very important. If you are assembling genes, as a single mismatch can result in the non-expression of the protein. And finally, we have the fusion polymerase, which is a proofreading polymerase. As you must be aware from your class on the polymerase chain reaction, we have two types of DNA polymerases: proofreading polymerases, which will correct errors in the base pairing of the DNA molecule, and non-proofreading, which do not correct errors. So these are the three enzymes which are utilized in Gibson assembly. The fusion polymerase is basically high fidelity, which means it reproduces the DNA exactly as it is. It allows a long range of amplification in the range of more than 7,000 bases. So this permits you to assemble large genes. It has a high processivity, which implies that it assembles DNA molecules with a high rate. So you have 15 to 40 seconds per kilobase. So that means if you had a gene which is about 7,000 bases long, it will require around 280 seconds to basically process that particular gene. That means your extension time would be in that range. It has a DNA polymerase activity, which means that it builds up the DNA from the 5' to the 3' end, and it also proofreads from the 3' to the 5' end. So you can think of it as a proofreader. So it's like the same enzyme is involved in writing the book, and the same enzyme is involved in correcting the mistakes in that book itself. So this is an analogy of the dual functionality of a fusion polymerase or a proofreading polymerase. The exonuclease, as I have mentioned earlier, degrades DS or double-stranded DNA in a 5', 3' direction at 50 degrees centigrade. Now this exonuclease is thermostable at 50 degrees centigrade, and it also generates 5' and 3' overhangs as a result of the degradation activity. I will refer to the specific diagrams as we go along. Finally, the third enzyme is the taq ligase, which catalyzes the formation of a phosphodiester bond between the juxtaposed 5' phosphate and 3' hydroxy terminal of two adjacent oligonucleotides which are hybridized to a complementary target DNA. So what taq ligase essentially does is joins two DNA molecules which have overlapping ends, and owing to its capability of thermostability, the reaction can be carried out at 45 to 65 degrees centigrade, and it will not function even if there is a single SNP or a single nucleotide polymorphism. Now what we have to look at is the manner in which these enzymes basically function. So one of the constraints is temperature. As the reaction is carried out in the range of 45 to 65 degrees Celsius, all the three enzymes should function at this temperature, as the reaction is carried out in a single tube. However, if you look at conventional DNA ligases, they are not thermostable. They will only function at low temperatures. Taq ligase is unique because it has been derived from Thermus aquaticus, which is a thermostable organism at high temperatures. It is a thermophile. And you have the T5 exonucleases. Okay, so let's look at the Gibson assembly in detail, and this is the overview, and I will look at it in the next slide. Okay, now what happens in Gibson assembly is we have two PCR products. This PCR product, which is in green, represents the promoter, and this PCR product, which is in red, represents the gene of interest. However, in order to fuse these two PCR products, it is very essential, or it's important, that we have this overhang region. The overhang region is basically a region of sequence similarity. Now you can generate this by using oligonucleotides. So you can carry out a PCR reaction using two primers and generate a promoter sequence, and then you can generate a gene. Now what's very important is that you have incorporated this particular common region in both these genes of interest. So you have the 5' N, the 3' N, the 5' N, and the 3' N, and you have this commonality. However, if you are supposed to invert this gene, if you wanted to invert this, for instance, you can shift this common region over here and you'll have a flipping of this particular gene. So the orientation of the gene is determined by this overhang over here. So when you conduct this experiment in the laboratory, you generate 2 PCR products and you try to assemble them using Gibson assembly. So Gibson assembly commences with step 1, and we introduce the enzyme T5 exonuclease, and T5 exonuclease will basically chew back at the ends. So the term chew back refers to the degradation of this end. So it will chew back these ends, these ends. So what you will have is overhangs. So you have an overhang here and an overhang here, and the general principle is when you have an overhang and you add a ligase, you will have an assembly at the overhang, as in all ligation reactions. Now you have your Gibson assembly step 2. Once you have your PCR products with their overhang, you have to add the enzyme Taq ligase. And when you add Taq ligase, there will be an overlap, and this overlap will result in the fusion of these 2 parts. Now what you have to take into account is the fact that there are certain gaps over here, and we'll move to that gap filling in the 3rd step. So Taq ligase will function at 50 degrees Celsius. And finally, we move on to step 3, which is the addition of the enzyme fusion polymerase, which will repair the gaps, and because of its proofreading activity, it will ensure that there is no scar formation in the gene. Now this is very important because if you have a single nucleotide polymorphism in any of these genes, there will be no expression of the protein itself, and this will represent a failed construct. So it's very important to utilize the high fidelity proofreading enzymes at this stage. Finally, you have your assembled gene, and this gene can be now subject to downstream processing. It can be cloned into an expression system, and you can express the gene itself, or you can also utilize it for functional studies in other model organisms. Now, in addition to Gibson assembly, we do have another procedure which is known as assembly PCR, and you have to utilize a software known as gene to oligo. Now this is an older method, and the method has basically been developed in a time at which there were no synthetic gene systems. So this method is no longer utilized. However, you should know that you can utilize this method in the event that you do not have access to specific genetic elements. Today, the system is basically focusing on gene synthesis. So if you want a gene of interest, you can order it directly by specifying the genetic elements, and a company or a gene synthesizer will synthesize that gene for you, and you will clone it and express it in your organism and vector of choice. Okay, so for the gene to oligo system, all you need to do is upload the FASTA sequence into the server, and you will have an output. You synthesize the oligonucleotides, you mix all of them together in a PCR tube, and you assemble using the assembly PCR protocol. Now, in the case of this approach, which is assembly PCR, you may have a lot of scarring or misrepresentation of genes, as the oligonucleotides can combine in more than one way. Okay, so this is the template DNA. So we will, for instance, this is from Daniel Radio. We want to basically express this template. So the first step will be identifying that particular template and downloading it from the NCBI. The next step will be to copy and paste the sequence into the gene to oligo program, and the gene to oligo program will generate these particular sets of primers. You then have to contact a synthesis company. You synthesize these particular primers, and then you have to carry out an assembly PCR under the following conditions. Or you may also carry out a ligase chain reaction. After you carry out the ligase chain reaction, you have to carry out a secondary PCR of the assembled gene, and then you can clone it and express it in a expression system. Now, the reason why this method is not utilized in today's ecosystem, in the GMO or the genetic modification ecosystem, is because the assembly itself is not accurate and the procedure is time consuming. Today we have evolved new methods for genetic engineering, and these are primarily associated with Gibson assembly and the engineering of synthetic genes. So the method which was generally followed traditionally has been rendered redundant with the evolution of new technologies. However, as a molecular biologist, you should be aware of this particular technique. So this brings us to the end of this particular lecture on synthetic biology. To summarize, synthetic biology is an emerging field of biotechnology which utilizes genes in terms of their functionality. So you can imagine synthetic biology as an engineering aspect of biology. And just as in engineering you have parts, for instance in electronics you have the transistors, resistors, the capacitors. In the case of automobiles you have your automobile engines, the gearbox, the tires, and so on and so forth. In synthetic biology we basically have the parts, and these parts are the regulatory elements such as the promoters, the genes of interest themselves, and the other elements such as the riboswitches and the terminators. Now, the reason why synthetic biology has gained importance in today's biotechnology ecosystem is because you can utilize synthetic biology to construct organisms or systems which do not exist in nature, and this forms the basis for industry, as these novel innovations can be patented. Now, synthetic biology basically relies on assembly of component parts, and this assembly is carried out using Gibson assembly protocols. And in order to develop new innovations in synthetic biology, it's very important that you understand the role of each and every component of the system. So in order to evolve into a synthetic biologist yourself, you have to understand the role of promoters, regulatory elements, terminators, genes, the different components of a protein, and the different terminal ends of a protein. So these are things which you must understand in order to develop a career or a profession as a synthetic biologist. Now, the reason why we have incorporated this particular lecture module in your lecture notes is because it forms a component of almost all aspects of genetic engineering. You will be utilizing synthetic genes to develop expression systems in model organisms, as well as to industrially upscale vaccines, as well as the production of recombinant proteins. Now, in Gibson assembly, you utilize enzymes to perform reactions and obtain synthetic gene constructs. In addition to Gibson assembly, you can also utilize assembly PCR, although it is not recommended in this particular age and time, as the technology has evolved rapidly to address the components and the assemblies themselves. So that brings us to the end of this particular lecture on synthetic biology. This is the last lecture in your lecture series, and we will be ending this lecture, and you will have to complete the midterm exam online, followed by the transition to the next 7 weeks of this particular course. Thank you very much for your attention, and I look forward to receiving your feedback and comments in the feedback section of the learning management system. We will also have a class which will be face-to-face upon completion of this particular lockdown. Thank you very much for attending this lecture.